My name is Professor Erica Fletcher and I'm from the Department of Anatomy and Neuroscience at the University of Melbourne, and I've been leading the basic fundamental research program for the LEAD trial. So that is understanding how the laser works in preclinical models and then translating that to humans. So 2RT, or retinal rejuvenation therapy, refers to a completely new laser treatment that's been used for early stages of age-related macular degeneration. And what's new about this treatment is that this laser fires very short, small packets of energy into the eye and changes cells at the back of the eye. And by doing that, it removes the signs of early stages of age-related macular degeneration. The mechanism by which the 2RT laser works is extremely interesting. So we have these small packets of light energy being shone at the back of the eye, and what happens is it selectively removes particular cell types that lie in the very back of a person's eye, called the retinal pigment epithelium. So these cells, these retinal pigment epithelial cells, get removed by the laser, and then as those cells rejuvenate, as they heal, we have lots of changes that go on at the back of the eye that causes all of the loss of signs of age-related macular degeneration. The 2RT induces a healing response, and the way that works is that the retinal pigment epithelium, which is a very special supportive cell layer behind the back of the eye, these cells get removed, and as they heal, they do two very, very important things. Firstly, as these cells heal, they actually divide. And that means that the new cells that are there are very young cells, and they are going to function like in a young person's eye. And then secondly, we have a wound healing response that causes gene changes at the back of the eye that causes a thinning of a very small membrane at the back of the eye. And this membrane, called Bruch's membrane, is critical for impeding and moving nutrients from the blood supply that lies underneath that membrane into the retina. So if we can improve the Bruch's membrane, that small thin membrane, we can improve that by this laser. We're actually improving how nutrients flow into the retina. So the 2RT has a very unique beam profile, and that means the way the laser goes into the back of the eye is highly unique. It's as if you could imagine a laser being made up of very fine strands, like in a bag of spaghetti, where each of those is in turn doing something at the back of the eye. And so what happens is, instead of just getting an enormous amount of cells just suddenly disappearing, what we have are only small numbers of cells disappearing at the back of the eye in a very, very unique pattern. That, in combination with the fact that it's a very short pulses, nanosecond pulses that are fired at the back of the eye, means that you have a very unique wound healing response. And very importantly, it causes no damage in the neural retina, and that's completely novel in comparison to any other laser treatment that is used in the eye. The 2RT protects rods and cones, and indeed all neurons in the retina, by being so selective. And what I mean by that is that this laser, because it only involves short pulses of energy, it only removes one cell type at the back of the eye, called these retinal pigment epithelial cells. And what that means is that there's no bystander damage in overlying cells. So unlike any other laser treatment where you get a lot of secondary damage, usually due to thermal damage from the laser, this particular laser sees none of that. The neural retina remains intact, completely normal and completely functional. And that's a very, very important distinction between the nanosecond laser, the 2RT laser, and any other conventional laser that is used in ophthalmology.